Welcome, welcome. Let's bring on Professor Anduza. For those coming on in, it is our Wacom Wednesday. We're doing a little bit of science, art, and chatting. And now we're going to bring on Danny. We've got a lot of cool science to talk with y'all about today. We have new dinosaurs, new kinds of classification systems. We've got memory, and we have a weird, although really cool study on speciation. Where it's actually been demonstrated how the two species of fruit fly may have actually speciated, even down to the neuronal level, which I always think is really, really cool. Let me bring on Danny. There we go. Invite sent. Let's hope today, chat. Maybe we'll see if we have any silliness with guest star, or if we'll mostly be working today. We'll see. We never know. Always a surprise. All right. Hey, Danny, how's it going?
Hey, it's going well, Belinda. How are you doing?
I'm good. How was the stream today?
It was good. Yeah, it was really good. Had some fun discussion, had some excellent raids, and yeah, it was nice to be able to get back into some science discussion stuff after our big stream anniversary yesterday.
Congratulations again on 4 years. That's huge.
Thank you. Holy cow. Yeah, it's been such a privilege to be able to do science outreach on this channel for the past 4 years. And big props, big thank you to members of the community who made that possible with their support. So thank you for that.
Hey,
I'm excited to be here.
you've been changing lives for 4 years now.
I appreciate you saying so. It made me blush. Shoot.
Before we get started, Danny, would you mind cranking the volume again just a little? I'm so sorry every time on the mic side.
Is that better? How's that?
Better, chat? Andrea, better?
What do you think?
I think it sounds better to me. Sounds cleaner.
Okay. Cool.
Better. Okay. Hi, Oscar.
Anyway, how has your stream been going, Belinda?
Good. We've been working on some science art inspired by Nintendo, and the Star Fox
Nice.
video game character as our Aspergillus fungus because it's goofy. So why not? Um,
Is he doing a barrel roll?
no, no, not. Although we should do
That's
one
the
probably.
only thing I know about that game. Yeah.
Is that where it's
Um,
from?
I think so,
See,
right?
I don't.
Back me up here, chat.
I
Confirm or deny. Do a barrel roll.
I've only know about Star Fox because of that Super Smash Brothers. I've never actually played any of the Star Fox games.
Gotcha.
But
Yeah.
Fishman and Armor Prayer says yes, so I guess we're on the right track of knowing where it belongs.
All right. Cool. Cool.
It is for that one. Okay. No. See, I didn't even know Armor Prayer. I was like, he shoots a green laser. That's cool. So he can shoot a green laser
Nice.
here too.
And he flies an X-wing starfighter, right?
Yeah. Oh yeah. Well,
Do a barrel roll. Close enough.
see, I always thought the barrel roll meant that he was supposed to jump or something like that.
I think it's
I-
like to, because in an aircraft you've got pitch, you've got yaw, and you've got roll, and so I think that's what it's in reference to. Is yeah.
That makes much more sense than than just okay. Yeah.
Yeah.
When
Yeah.
I give you a knuckle, a sandwich of
It's fact, Simon
science.
Mark, indeed. Yeah. Um, so yeah, we had a chat. Danny, did you tell the party people we had a cool chat today?
I did not actually. No. Yeah, we did have a cool chat today, everybody.
Just mentioning to folks, we're working on a potential application for a panel for TwitchCon, bring more science to everyone's ears, and everyone's really excited about it. So we're fingers crossed, y'all. We get a couple of science events this year at TwitchCon.
Yep.
We have that submission for a DNA extraction live experiment for folks to do. We might have a science
so
panel
cool,
too. I think collectively we can really demonstrate that Twitch isn't just for games, but for easy for education to take hold on there.
Yep. Yeah. Oh man, I'm excited. And now that I've been to TwitchCon last year, I did my first TwitchCon. I know what it's like. I feel like really cool stuff this year. Now that I've had that boots on the ground experience, so excited
Yeah.
about
And Chad, we
Yeah.
started like early discussions maybe of having a meetup too. So if you're coming, might have like a big meetup for all the fans of like the sciences and educational streamers who are coming to bring everyone together who maybe, weren't at one of the events, but we can have like our own event. And like actually there's a really nice dock, really beautiful boat dock right behind the convention center.
Oh, cool.
Yogurt Girl and I accidentally discovered it last year. We just walked behind the center and there's all these yachts and it's like a boat deck and there's like I think that'd be a really cool place to go hang out there too. And then there's like food right near. It's like, ooh, there's a lot of
Or
cool
if
stuff.
anyone is arriving via ocean liner or submarine, that'd be an easy way that they could get to TwitchCon.
I mean, that would actually be really incredible. Can you imagine? Step off the dock, like, all right, going to the con.
That would be pretty cool. Yeah, yeah. Just hand the submarine valet your keys and you're off to TwitchCon.
Exactly, yeah. We also showed off, I don't think you've seen this thing, Danny, yet, but we got one of these in the mail. So, these are, it's a set of STEM toys that are Star Wars brand, and it's all kinds of slimes. It's got little LED lights in there, and Lita and I wrote up all the details, like all the science behind each of these things. So each one has a collector's card and like different color science and whatnot. They put out a whole line of Star Wars toys where we did the write-ups on it, and they released it.
That's awesome.
For May the 4th. It was released, and so there's all these press releases that went live, and we couldn't talk about it because we had so many Disney NDAs that we had to sign. But
sure, yeah.
But now it's like all out of the bag, and it's like all of our little write-ups, and we're even in the press release. And I was like, oh man, that's so cool.
That's really exciting. Yeah, holy cow! When will it hit store shelves? When can people get it?
Already. So it
really?
Yep, it went live on May 4th. So like Amazon, Target, Walmart, and what's really nice is it's really affordable. Like relatively speaking to how much toys cost nowadays.
Yeah.
But it's like, so here are some of the products. Here, I'll shrink it down so y'all can see. Yeah, and we don't, just full disclosure for anyone, we don't get a commission of any of this. It's just we did the consulting.
Well, if it does well, then could get rehired for that. Yeah.
We have a second job that we finished as well that we are also under NDAs for, but hopefully we'll be back for more. But yeah, there's all kinds of these like different putties and like building and digging, and there's when I was a kid they had the equivalent of you can dig out like a dinosaur bone, but here it's a gem dig. So it's like there's different gemstones and geology that you can learn, and you pretend to be the armorer from the Mandalorian, and so
cool.
And what was I was really excited that the CEO guy wrote, applying Horizon Group's innovative approach to the Star Wars brand, backed by the educational content developed by research scientists Drs. Balint and Lita Cacho, has enabled us to make science subjects more accessible to fans of all ages.
Very cool. Holy cow, that is awesome.
So I was like, that is...
Congratulations.
Thanks. Yeah, there was like 20 different news outlets that reported on it. That's the one from the Associated Press. I threw in the link if anyone's interested, but I was like, whoa, this is... It's like next level.
That's really exciting.
So we were excited. We're like, hey, maybe Twitch will be like, that could be a good ambassador right there. They did consulting and put out Star Wars STEM toys. They might be like actually into this science thing.
That's awesome. That's really awesome. Holy cow. Cool stuff, Balint. Holy moly.
Hey, it's still... I'm just still blown away, too, by yesterday how much fun it was for the 4-year party. Just as an aside, how cool it was, like, to see, like, old clips and almost, like, the growing up of the channel. And because I never knew you as a small starting up streamer. I've always seen you as like this, like the big league streamer of SciComm, like the pillar, like I think I told you, right? Like we ended up on a random raid into your stream, and it was like, all right, well I'll drop a 6-month sub on this guy because he's amazing and I love dinosaurs. But yeah, that was like 2021, I guess. Yeah. So you had already.
Really? So I would have been streaming for like maybe 9 months or something at that point.
You were yeah. You were already partner.
Oh,
and,
okay. So that would have been after March of 2021.
Yeah. And it was just
Okay.
like, well, this guy is great. This is where like someone should aim to be if they're doing science. And it was really cool and just like humbling to see like where, that it has been 4 years and like the community's just grown and how you've been able to go from, the office stream to field work to museum visits, and it was a super cool experience to be able to see that. And chat, please follow Danny because I tell you, in 4 years he's gonna be running Twitch.
Don't say that. Oh boy. Don't curse me with that. I'm not a corporate guy. I appreciate that, Belinda. Holy cow.
It was again
we've
just
got some exciting stuff planned for this summer. Holy cow! I was just talking about the Utah Raptor block stream that I'm going to try and do with Jim Kirkland. Did I tell you about that last time?
no.
So there's this big. It's the size of like a minivan or bigger. This block of rock that contains the bones of probably a dozen different Utah Raptor dinosaurs, adults, juveniles, and hatchlings. They seem to have died in quicksand. It's the first instance ever of dinosaurs getting stuck in quicksand that we know of. This was brought, the whole block was brought from the desert of eastern Utah to Salt Lake City, and it's just sitting there in the laboratory, and you can see all of these bones right there on the surface. Who knows how many more are there below the surface? But with any luck, I'll be able to do a live stream with Jim Kirkland, the state paleontologist of Utah. And we'll be looking at all these different bones and just talking about it. So pretty exciting stuff. It'll be a unique stream for sure.
Why has it not been opened up?
Lack of funding.
Okay,
Yeah.
so it's like they have the sample, and it's like if
But
you
this
had
is a decades-long project.
Really?
It's like, oh yeah, like we will continue working on this thing for decades. Because it's really slow-going work, like chipping away bit by little bit the rock there. It's really tough matrix, and the bone is also very fragile. And so you got to work really slowly, like under a dissection microscope with like tiny little, very, very, very precise tools, basically like needles in thin devices.
Wow.
That's the kind of a tool we use for this.
So
yeah.
I'm really naive then because I thought there would be some level of automation to this at this stage, like where it's not like done by hand, like because it almost sounds like it's like how you're doing it in the field, but instead of in the field, it's in the lab, and
it's much more slow than in the field. In the field, we got to get these things out of the ground ASAP. So we have limited time and limited money, and sometimes you have thunderstorms rolling in and stuff like that. But we're in the field, we're collecting the fossils with a bunch of rock around them. Basically, so we can do that carefully but quickly, and then get the bone and the rock back to the laboratory, and there you can do the slow, precise, painstaking work of what we call fossil preparation. That's when we're removing the matrix, the surrounding rock, from the bone. We're using tiny little dabs of glue to put pieces of the bone back on that may have fallen off. We're consolidating it with chemical consolidants, all kinds of stuff like that. It's really slow-going work, and it takes an extremely specific set of skills to be able to do that. It's not the sort of thing that you could teach a computer to do. It requires an artist or a sculptor's touch. In fact, a lot of fossil preparers have a background in art, especially sculpture. Because you got to think in terms of 3D like that. You're constantly solving these problems in your head. Like, okay, if this is a tiny little humerus of an hatchling Utah raptor, I expect it to be this shape, so I'm going to be just chipping away the rock only to this certain point until I get over here, that kind of thing. Constantly problem solve.
Wow. Yeah, because, I mean, and it's like you can't be a bad 3D artist then either, right? Because...
No.
Right, because, I've seen, 3D art where it's if you look at it from any side, it looks great, but if you take it as an entire piece of 3D, it's there's everything it looks like was individually done as a flat surface rather than thinking of it as one unique piece. I don't know why it never registered in my head that it's such a beast to be able to do in the lab like that. That's really, really cool.
Oh, yeah.
There
are very few people in the world who can do that really well.
Is there an image of this Utah Raptor pile.
Yeah, if you just look up Utah Raptor block, you'll find it.
It sounds so cool.
You can find YouTube videos on it too. There was a GoFundMe at one point, like trying to crowdfund being able to pay a preparator, someone who has the skills to be able to do this. Scott Matson was working on it for a few years, and they just like ran out of money basically. So we might be trying to raise some funds for it with this live stream. I got to talk to Jim about that and see what his feelings are. But I think it could attract a lot of viewers. Like, people would be really excited to see this. It's going to be a pretty unique, interesting thing.
It's amazing how much information could be trapped there as well, because you could.
Oh yeah.
Like, presumably get information about their social structure, the development, the timing of things, and if he's into, and I can't remember if you said about the Jim, but if he's into cutting into the bone, yeah.
Oh yeah.
Then you can get a.
There are different groups of theropod workers, people who work on meat-eating dinosaurs, who are all looking at this thing and just absolutely salivating. This is like, it is unique. There is no other assemblage of theropod dinosaur fossils like this anywhere else in the world. It is one of a kind.
Wow.
And already we have disagreements about, are these all Utah Raptor, hatchlings and juveniles and adults, or are there 6 different species of theropod dinosaur in there that all, you've got a small species of dromaeosaur and a medium one and a large one. There's already disagreements about that between different theropod researchers. That'll be one of the things that we cover on this stream.
I wonder if you'd be able to re-lump things based on this evidence, right? Like, if you find ones that are small, and they match the record of something else that had been classified as something like a different species, right? But then if you have them all here, maybe you're more likely to be like, well, no, they're all part of the same family unit.
Well, or if you cut them and you determine that, like, oh yeah, the small one, it's not an adult, it's a juvenile. It's just a juvenile Utah Raptor. Like, that could be really helpful. Yeah.
That's going to be such a cool stream. That's so cool.
I'm pretty excited about that. Yeah.
Golganak was wondering, Dan, if people can still donate to the preparation. Golganak, do you mean this one or for Danny's trip?
I think she means the fossil prep for, let's see, let me look it up. Utah Raptor Project, crowdfunding. Here's a link to the Utah Geological Survey website in the chat. And I don't know if they're still doing a fundraiser there. They may have shut that down. But that's like your main clearinghouse. You've got that website up. Perfect. But that's like the main clearinghouse for information there, is on the UGS website.
Gotcha. I see, Golganak, I was thinking of the Amazon link for Danny, which I'm like trying desperately to find in my messages. I'm like,
oh, don't worry about it. Yeah.
There was like one item left, and I'm like, I just want to make sure that we get all the items like taken care of. Just to make.
Smartfree wants to know help. Oh, go ahead.
Oh, just to like show Jim, like, yeah, we got this.
Oh, I appreciate that. Yeah. Smartfee wants to know how big a living, fully grown Utah Raptor would be. About 20 feet long. So, like, it's about a polar bear-sized animal, but longer than a polar bear because it's got that long tail. It's a big animal. It's a big animal.
Is that size represented in any of the media outlets that are reporting on Utah Raptor?
Sure. Yeah.
Okay,
yeah.
Cool.
It's picture it like the velociraptors from Jurassic Park, but bigger. Like, significantly bigger.
Gotcha.
Maybe close to twice as big, honestly.
Oh, wow, okay. Nice. That's huge.
Yeah. Yeah, it's a big animal. Like, it's as long as a pickup truck. Yeah. Bigger than Sweetie Pie here, certainly. Yeah.
Which pie was with you earlier today? I forget.
That was Mini Pie was causing trouble. Yeah. We had to lock her out of the room because I don't know, she went nuts over those treats. And she knew that there were more. I wasn't taking them out of the small bag. I've got a big container now sent to me by a community member, and Mini Pie knew it. She's like, I want more of them. So
Munchkin
yeah,
was enjoying over her dinner. She was watching you play with the cat, and she kept pointing at the
really.
cat. So she was enjoying the shenanigans that was being caused.
Well, lovely. I'm glad to hear that.
She
Excellent.
she knows 3 streamers' voices: yours, Irish John's, and Thug Shell's voices. Where she'll like
It's a good
actually
company to be in.
hone
Wow,
in on
I'm flattered.
the stream.
Yeah. Yeah.
And it,
Awesome.
dinosaurs. I mean, come on. Chat,
Yeah.
as I posted into the chat, and I'll also go ahead and pin the message. This is for the link for the last bit of items on the Amazon wish list to support Danny on the field excursion this year. There's 2 items left. Can we buy out everything, folks?
Let's see. Shoot.
I think we can. And Chad, they've had to add more because they kept stuff getting bought out.
Repeatedly, which is excellent. Yeah. Ethan, who's the kind of our leader for this whole project, this was his brainchild. He's been super impressed by the level of generosity displayed by this community, and he's almost a little embarrassed. He's like, we're going to be one of the best equipped paleontological crews in the country, if not in the world. This is incredible. So, we're pretty happy.
It's a sad commentary. I mean, it's amazing that the community did this, but I feel like it's a sad commentary that the community is the one who has to rally, and there's not scientific funding available to the researchers and getting you what you need to do the science well, right? Like, it's just that's a little bit disheartening that it's amazing that it can be crowdfunded, but at the same time, it's like it shouldn't have to be. You shouldn't have to be dependent on that.
For sure. Yeah. It would be nice if we had more funding from, I don't know, say the federal government or something like that. Speaking of which, let me send you a link real quick. This is a tweet that I saw today that I thought was funny and instructive. So this is a dinosaur being given as a gift from one world leader to another. It is a model of Anchiornis, which is a dinosaur that comes up in my stream a lot. It's a very bird-like dinosaur. But it's being given from the president of China to the president of France as like a token of goodwill. It's like, here's something they're super proud of in China. Like, the origin of birds. Most of that information from the past 30 years has come out of China. And so it's like a real point of national pride there. And they're doing a tremendous job of like building new museums and funding research, making sure that there are like, there's a new generation of paleontologists who are being taught there. And here that is like on the world stage. I just thought this was pretty cool. I'd love to see more stuff like this. I don't know. Sometimes I wish we had this kind of attitude here in the States,
That's huge. That's absolutely huge.
Yeah.
Isn't
that cool?
Yeah.
Yeah.
Oh, and
What's funny is in the original news article, the person captioning the photo didn't know what it was. It's like, oh yeah, there's some sort of ornate bird model that was being gifted to President Macron. It's like, no, that's Anchiornis. That's a dinosaur right there.
That's super cool. It's like there is that that's that scientific information behind it.
Pride, so proud of their achievement. It's super, super cool. And it's neat as a paleontologist to see that kind of enthusiasm.
I wanted to show you a piece of art that was actually inspired, now that I'm thinking about it, by you in our community. Our friend Tatum did this. And I think maybe Fishman did something as well. Fishman, let me know where I should be looking. Because I'm not entirely sure where you want me to refresh, Fishman. But it's not finished yet. But this was inspired by you.
Oh, yeah, very cool. I love that. That's really neat.
So, it's the your inner dinosaur. Oh, Danny's list, Fishman. All right, hold on. That's what Fishman said. All right. Well, Danny, you're done.
Holy cow. Oh, my goodness.
Fishman, thank you so much for contributing to Danny's list. So that was Fishman grabbed the last 2 items.
I'm gonna text Ethan right now and let him know and see if we get a response. Um, but that is pretty exciting right there. Holy cow! Thank you everybody for your generosity.
Yeah, and
That
Fishman,
is
thank you for
amazing.
topping it off, Fishman. And yeah, the last just 2 items. Thank you so much, Fishman. That's huge. I love seeing that the community, banding together and supporting you on that. Yeah, it's absolutely incredible. I know we've been pushing it to like on our Discord and during streams. We're like, come on, everybody, support Professor Anduza.
um, Pretty exciting stuff. Yeah, holy cow! And it's coming up too. He'll be out in the field in like less than 3 weeks at this point. Um, or maybe a little. Yeah, I will be leaving for the field in less than 3 weeks. I'll be out there in Utah before we go to Wyoming. Yeah, cannot wait. Now, Ethan just sent me a reply with some fireworks emojis. So
Nice.
yeah.
That's so cool, and it's just like, yeah, we got everything, and like you said, there's a fair bit of stuff on here, and like you've said too, it can be used for many. Like this isn't just for one set of field work. Like you can keep using this, and that's I think
Some
really
of those
really
tools
cool.
might outlive us in a good way. Not that we're gonna have short lives, but like us as a paleontological crew, there will be hopefully future generations of paleontologists who will be using some of these tools in the future. Um, so pretty thrilled about that.
Yeah, I was gonna say you're not gonna be like a dinosaur, right, Danny? Living fast and short.
No, hopefully, I don't know. It'd be nice to reach a ripe old age, maybe.
I just
But yeah,
the phrase as you say of like of lit that
dinosaurs lived fast and died young.
yeah, it's just like that's been really resonating with me, and the more I think about it, of like how little information we have about development, about timing, about life history, really, and how much information there could be gotten from like this, like the giant like Utah Raptor rock, and just a lot of these where I was like, wow, that is really cool stuff. That is like every day I'm just like, wow, this is amazing work. And again, thank you so much for sharing your knowledge with all of us and chatting about it. And
thanks for being so enthusiastic about it, Belinda. Holy cow! Yeah. And thanks for all of your support, getting the word out about the wish list and everything else. This is stellar stuff. So thank you for your support, everybody. Thank you, Belinda. Holy cow!
And we're always behind you, no matter whatever you need, whenever you need. We're always right there.
Hey, likewise, as you continue to do your science outreach here, whatever I can do to help out, however we can support each other,
It's the importance of realizing that it's not really competition, it's for the betterment of both of us, as everyone's going up together in science and education on the platform, making, opportunities for others, and I think that's the magic of it, yeah.
A rising science tide on Twitch raises all science boats. So
exactly. Yeah. All of the scientists, let's band together. Let's do it.
Yeah. Science solidarity. Yeah. Yeah.
Speaking of, should we do some science solidarity? So, what would you like to chat about tonight first?
Yeah. Which of your papers were you more excited to discuss? Because I want to make sure that we cover that. We only talked about my paper last week.
Yeah, but that was one of the best, I think, collabs that we've ever had, because there was so much and I'm not putting down any of the other ones, but it was the perfect paper to have the overlap on, right? Yep. Because it was the neuroscience side of things, the paleontological side of things. There was controversy. It was just so cool to chat about. So in terms of the paper, so Chad, I have one on molecular memory and how cold affects molecular memory, and then another one that is much more evolutionary, where it's trying to recognize the evolutionary breaking point of perhaps a speciation event.
This is the singing fruit flies, right?
Yeah.
I'm kind of more excited to talk about that one, if that's what's...
It's your call, sir.
Let's do that. That sounds great. I'm always game to talk about speciation and, how new species form.
And just so folks have it, so there's the actual paper is behind a paywall. However, what I really like that these authors did is they uploaded the paper to bioRxiv first. And so that's freely available. And what's really neat is you're supposed to, there might be some differences here. This should be pretty close to the final published, published actual paper there. Which is, it's really nice that, there should be a link between the two. And then, yeah, Dr. A said for chemistry, there's our archive as well. So preprints get uploaded. This has been peer-reviewed, y'all. Here is the actual peer-reviewed paper. It's in the journal Current Biology. And it's just, I thought it was really exciting because it's actually looking at and trying to figure out and analyze how different species evolve, but on a molecular level. And so, actually, I'm going to go ahead and I'll pull up for y'all just the actual paper because I think that one's a little bit prettier to see. So I dropped out the bioRxiv one, but here's the paper, and I really like that these studies have these graphical abstracts.
That is nice.
Yeah.
I wish we had more of that in paleo. Stuff that's been published in Current Biology. That does seem to be something that's kind of a trademark of this journal, right?
Yeah, and there's some journals where it's like, yeah, you have to have some flavor of graphical abstract, which is great because it shows some, like, you have to be able to communicate your science in, like, a very succinct image. And so I think this is really nice and straightforward where it's like, they're like, here is this TN1A neuron. Hi, Harissa, welcome in. Here is this neuron. Here is the ventral nerve cord. So it's essentially analogous to the brain stem in the fruit fly. And then it zooms in on their brain, and they're like, there's this neuron. And they have this little song note here to denote singing. Like, even, okay, they went that far to do. So that's in the Drosophila melanogaster species. That's your common household fruit fly. That's the fruit fly that everyone studies. In terms of speciation, so for different types of species, usually the only thing that's been studied is the sister species of Drosophila melanogaster, meaning that they are extremely close relatives where they can interbreed and make infertile next generation offspring. So this other animal that's being studied here, and actually there is a phylogenetic tree. We're going to jump ahead, y'all, just so I can illustrate this. This is the evolutionary gap here. Hi, Nerdowino, welcome in. There's Drosophila melanogaster, and this is Drosophila jacoba. So it does have a split point about 6 million years ago, which is when they split. Both are endemic to Africa. Well, actually, melanogaster now is worldwide. We actually suspect that it's spread and took over the world basically as an invasive species due to humans, actually.
Interesting.
So melanogaster is on every continent, almost every temperature zone. They're just all over the place, for better or for worse. They're just everywhere. Jacoba seems to be maintained in Africa, so these two species still coexist and they hang out. But there are, if you cohabitate them, they won't interbreed.
Interesting. Okay. I guess 6 million years of evolution, divergent evolution there might do that. Like they're hesitant to hybridize.
Yeah, and so there are sister species where, again, it's like with melanogaster, there's simulans, there's Seychellia, which are on the Seychelles Islands, and then there's also Merciana, which is on another island. Maltese, I think, is the island that they're on. And they can all interbreed. But the reason it turns out is that they are not fertile is because when their chromosomes pair during cell division, some have genes missing, and so they don't fully pair. So you'll have like a pairing event, but then like a big lump that comes off. And when that doesn't happen, your next generation just it can't develop. And what you actually end up seeing is if you dissect those ovaries, the ovaries are very thin and barely existing.
Gotcha. Okay.
And so why I thought this paper was cool is because the idea is that there might be other barriers to mating, not just physical barriers, as in these animals like are unable to mate because the parts don't connect, but they also sing. They also sing songs, which I thought maybe isn't something that chat knows.
You can have some kind of allopatric or sympatric speciation going on there, because they're just not, they're not feeling the vibes with the different songs.
Exactly.
It's interesting stuff. Yeah.
And so in the graphical abstract, it actually gets to be pretty straightforward. Of like, well, there's these neurons in Drosophila melanogaster, and Drosophila yacuba doesn't have these neurons. Turns out during development, they're pruned away, and then it doesn't do that part of the song. And so that's like the crux of the study, and I'll show y'all how they got to that. But it's figuring out that, hey, there's this evolutionary barrier to mating because of song, and then let's get to the neuronal level of how exactly that happened.
Actually tying the songs to the physical structure of the brain. That's really wild. That's beautiful and elegant, and that it's so clear-cut like that you can actually observe physical differences between the brains of these different critters that result in different songs. Like, that's fantastic. That's wonderful.
And it's... The reason I really like this paper is I'm a sucker for evolutionary biology, but I think a lot of times there's not enough work done on evolutionary biology from a molecular perspective, where it's just like you don't have the funding to do both, or if you do have the funding, you don't ask the questions of that you can as a molecular biologist. And so here it's like we have this observation, and actually the songs are very easily spaced out. So here is just an acoustic track of the two songs, and you can even by eye tell the difference. Yeah. There's these pulses in blue, and then there's this sine portion which is a little bit more narrow in yellow. And so you don't observe the sine pulse in yellow in Drosophila yacuba. Instead, you hear this clack, which is indicated by this giant acoustic peak and then a gap, and then a peak and then another gap. And what's really cool to me is that this is repeating. So like the timing of these in between different individuals is consistent.
Interesting. Very cool.
And so like even...
before we get into more detail on that, I gotta ask, how are these animals actually producing that sound? Is it their mouth parts? Is it their limbs? What's the physical mechanism for actually producing the song?
So a couple of different components. There's the tergites, and so that's like the little stripe functions along their abdomens. It's like pieces of their armor that instead of being a singular unit on their abdomen, it's pieces sticking together, and they can rub those pieces together as they curl their abdomen to make sound. And so that's one component. And then there's also a pivot of their bodies. So it's like it's physical movement. It's not by their mouth. It's they wiggle their wing, but they don't make the sound with just the wing. So it's almost like a hitting on their body and then curving their body around to make these kinds of sounds.
So those motions are producing vibrations.
Yeah.
Transmit the air sound.
And I can get an audio clip if y'all want to hear what this sounds like.
Yeah. Dave Karen says, "Do a body roll."
Yeah.
Nice callback there.
Let's see. All right, here we go.
And speaking of fruit flies, I have one in my office right now. Come here.
Take it out, Danny. So, this isn't a video from the same group, but the principles will still apply. And so, when you're doing this kind of recording, you have to have microphones from a couple of different angles, and so because we can't hear it with our own ears, and there's a lot of post-processing that ends up happening. But you can also have like high-speed videography, and what's really I think neat is there's a lab at Princeton that does this, and they've perfected the video settings. If you do it in color, there are some behaviors that cannot be detected by current color cameras because the frames per second and res are not high enough in color cams when compared to black and white.
Wow.
So like, just to give you a sense, y'all, of like, surely we figured out Fruit Fly song. Like, the tech isn't really up to date to be able to even figure out these
things. They're too advanced for our level of technology. I love that. Yeah.
Yeah, but this is just a short video, a little bit on how you do. You know, it's an example of sexual selection. They have the songs too. That low humming.
Yeah.
So that first part, the brrrr and extended, like almost sounding like that purr, as Harissa said, that's that sine function. And then, because it's, so if you look at the wave here, right, that's like all together in this orange. It's just like together. And then when there's pauses in between the sounds with those peaks, that's the pulse part of the song. And you can actually hear it once you know, almost, I mean, you have to, what's called deconvolute the data. You have to take the sound and process it so that our ears can hear it. But yeah, that's the beginning stages of that song. And it's the male, so just so y'all see here too, the male is here up on the top of the image, and he's approaching the female. The female can actually do many different things to dissuade the male. And what's really, it's always made to be a joke, and I think it's pretty funny, but it's also really interesting biology, is if a male gets rejected, there's a neuropeptide that's decreased in a part of the male's brain. It's called neuropeptide F. And when that goes down, the males seek out alcohol-laden food.
Really? Wow.
And there's always the, they're just like regular dudes, you know, comment. But what's really cool is that molecule, that neuropeptide F that goes down in their brains is the same molecule in humans that links addiction. And so in humans it's called neuropeptide Y, but it's the exact same thing in a fruit fly, and so that's how you can actually study addiction behavior in a fly and translate it to a human, because it's the same neuropeptide between the two things.
Is it thought to be homologous, or could it be convergence?
It's thought to be homologous.
So, chat, basically that means that, like, both we and fruit flies inherited that trait from our common ancestor, probably, like, 500 million years ago or something longer ago. This would have been before the Cambrian period. Back in the pre-Cambrian, maybe close to a billion years ago. That's wild.
If you look at the human genetics, there's a couple of additional of these. Like, there's been gene duplication events, but it's the same root.
Same gene.
Exactly. Yeah. Where it functions so well that you can take out the fly version and put it in one of the human homologs, and it works.
Yeah, that is really cool.
Yeah.
That's extremely basal. Yeah, absolutely. That's so neat.
Yeah. And so it's just it's not having to do with any of the song, but it's like a lot has been studied on male courtship and male like neuronal decision making based on this kind of mating behavior, and this, like, again, the overhead cam and having the acoustics put in.
Yeah.
And so Harissa, humans have this neuropeptide for addiction behavior. Female flies also have this neuropeptide. And actually, Lita's PhD was on decreasing that neuropeptide in female brains. It led to a similar thing where they sought out alcohol-laden food, but what was scary and cool was that the next generation inherited this decreased level of the neuropeptide. So the next exactly. So even though the next generation didn't have any kind of that original exposure, it inherited it as if there had been some kind of exposure.
Really interesting.
And it's all based on that neuropeptide. So, and Harissa, so then the question is, okay, because this molecule is also present in humans, is there addiction behavior that could be transmitted across generations in humans as well? And so that's a big stone throw. That's not proven, but like that could be a connecting point between those two. Yeah.
I can see how like that would be more easier to imagine in humans, like passing something like that along to your baby, because you know, in placental mammals like us, baby is held there in utero for a long time. There's a lot of, you know, exchange between the mother and the fetus and everything else. But for these critters who are just laying eggs, you wouldn't necessarily expect there to be that kind of heritability of stuff that happens within the life of the individual organism. So like you could more easily demonstrate that is like an epigenetic effect. Really cool.
And the one complication is with humans, right? We have all of our embryos at the start of life versus flies. And sleep well, Fishman. Thank you for the raid and for hanging with us and for contributing, finishing out Danny's wish list. Thank you again, Fishman, for that.
Really appreciate that, Fishman. Holy cow. Was that you, Fishman?
That was Fishman. He did both of them.
Fishman, be sure to email us at dinosaurcampaign@gmail.com if you want your username put on a little label on those items so that you can see those during the live streams. Yeah.
And that email, Fishman, is pinned to the top of the chat right now if you need it written down too. Or you can just DM me. I'll send you the info. I'm sending everyone the info.
Yeah.
But yeah, so in humans, you know, if there is this, like, the idea is if there's an maternal stress, all the embryos are going to get affected. And then that's kind of it, right? But in a fly, there are these stem cells, and they're dividing asynchronously, or asymmetrically, meaning that there's one cell stays a stem cell, the other cell becomes an egg, and it keeps going on and on. And so there might be embryos that if you do a stress, they'll be exposed to that stress. But then if you wait a week, you've cleared out all the embryos, and now you have brand new ones that were never exposed to it, even maternally. And it turns out, when Lita tested a week later, those embryos didn't inherit that effect because presumably they weren't even made at the start, and so it shows kind of transient epigenetics.
Interesting stuff. Wild.
Yeah.
Very cool.
And then Harissa, we don't know in humans, so this is very much like on the front lines on fly research. Knowing what it means in humans is very much a hand-wavy possibility right now. The molecule is present in mice. It's again studied in addiction behavior, not necessarily at transgenerational or cross-generation behavior. It's something that we can guess, and maybe it happens, but there's no concrete evidence in the mammalian or human system that is what's going on at this stage. It's a lot easier to figure out in a fly because its generational time is 10 days versus a mouse that's, 4 to 5 months, and more money and cost. And then humans, like, you're not testing at all, or even if you do test, how do you know what's an effect of almost an accident, right? Like environmental stressors that could be influencing it versus it is what you think it is. Like, human studies are notoriously difficult because we live lives. We're not in a clean room box the entire day. And
Thank goodness.
Yeah. Oh, yeah. So there's.
I don't know. Some people on Twitch might be, but hopefully that's by choice.
Exactly. Hopefully it's by choice. Here's a little bit more of the song if y'all are interested in, again, hearing a little bit more.
Very cool.
And so what I think is really neat on these is that in the past, before this paper, there's precedence for if you turn on particular neurons with a shining on a light on the fly brain, it'll trigger them singing this song. So you can trigger it independent of the stimulus. So that's
so cool.
That's a little bit of what we already have as like a basal amount of information of, like, we know in a melanogaster these are the neurons that are important, and what you can do is, and you can hook this up to any neuron, where if you shine a light on it, on the neuron, it'll release calcium, and that'll activate the neuron. And so it's called photoconversion. So you have this inactive state, you shine a certain wavelength of light on it that you would never have like the fly is never going to experience this wavelength of light in
Sure.
because it's like 405 nanometers or 532 depending on what you put in, and that's not visible spectrum. So you get to shine it on, you get to activate the neuron, and it by opening up calcium channels, and that's what produces the firing of the neuron, and you get the behavior. And that to me is like the crux of genetics, right? Like if I turn off this neuron and they no longer do mating behavior, that shows it's necessary. But if I can turn it on in the absence of the stimulus,
that shows it's sufficient.
And then you're set.
Yeah.
That's the
Very
big
cool.
push, yeah. And so here I thought it was really exciting. It was like, well, this Drosophila cuba, by using these same kind of acoustics, we're like, okay, we can't hear anything. Well, so then what's going on? Like, why is there none of this sound? And it's been known that they have this clack sound. This isn't the group out of Princeton that's been doing this, but they have observed that there is this clack sound, and these two sister species, Drosophila cuba and Santamea, have the clack. And what's really cool too is if you look on the tree, this Tesserae and Drosophila erecta, which are like those relatives there, they still have the sign and the pulse. And so it's just in this group
Neat.
where you've lost the sign and you gain this clack sound. And so again, it's like, it's just so cool to look at the tree and be like, it's so clean. Like, these creatures lost something and gained something else to replace it, and no one else flanking them has this.
That's very, very cool. And this gets into something we were talking about on my stream toward the beginning today, was molecular phylogenies, and how important that was when that spawned a whole new field of science, basically, being able to create these family trees just based on molecular data. And now that is super important for stuff like this, like that foundational kind of work, hopefully to this study right here in the year 2024. That's awesome. Yeah.
And I think, although as a grain of salt, it's still difficult to fully interpret the data, right, by its own, because there's still like the thresholds for what a different species is. Like, it's kind of a mess.
Typical phylogenetic work. Let's start coding some fossils where everything's so subjective.
Well, and
but that's a nice reality check for me though, because I always assumed like, it's all A's, G's, C's, and T's. It should be really easy to come up with a phylogeny with zero ambiguity, but it's apparently not.
What's really frustrating to me, Danny, is like on our, we do the new species of the week every Friday, right? And some of them have different thresholds for saying what is sufficiently different to call as two different species. And so like, well, then what do you do? If even if the papers that are citing each other are doing it slightly differently, like that's a unique one. And then like I think it gets more easier with like how you're saying with the A's, T's, C's, and G's if you're looking at it on a genetic level. So sometimes you can look at a single gene and make one of these trees and link it to the species and saying like, okay, here is where this species falls into the tree for just this gene.
Oh shoot! So they're not doing whole genome phylogenies. It's just based on single genes.
Depends. Yeah. Sometimes it's just like, here is a tree, and looking at the evolution of this one gene. This right here is a global image that they're pushing, but it's you see a little bit of both, and it kind of. I guess it makes sense based on what your question is, right? If you're looking at the evolution of a single gene, it almost gets a little bit easier to make your argument of like, if you're using this gene as a delineating line between this species and this species, your tree looks a little bit cleaner.
Gotcha.
But
well, let me ask you this though, and it might be a naive question, probably will be a naive question. That's why I'm asking it. Could you ever get fooled by convergence in a molecular phylogeny? Could you ever have two different genes that evolve separately to look extremely similar, or are genes just too complex, and would that be like too much of a extremely unlikely event?
So it's like if the actual coding sequence is very similar, and so two things that would be more distant are now going to look closer just because you're only looking at that coding sequence.
Yeah, that's what I'm asking. Yeah.
So the way that is a concern, because it can happen, right? Like if you're, well, because if your gene product, right, is like, you're making a few amino acids and you're putting them together to make a protein, there are really short things like neuropeptides are classically on the much shorter side. And so that increases the likelihood that something's going to be very similar. And so the way you tend to overcome that is to include the entire gene body. And that includes the exons, which code for the protein. And then there's the intronic region, which is the regulatory sequence. And usually those regulatory sequences under higher rates of evolution than the protein coding ones. And so that's how you would overcome it, is that those are not going to look similar.
That makes sense. That makes a lot of sense.
But yeah, no, it's totally a thing. Like, you can't just look at the protein. Because, like, the neat thing about protein coding, right, is that its 3 base pairs make an amino acid. And a lot of times, if you don't have the correct 3 variable sequence, it can still make the same amino acid. And so it's the difference between a synonymous mutation that makes the same thing or non-synonymous. And actually, like, if you have a synonymous mutation, that could be variants within a population even.
Gotcha.
Because it makes the same thing, it's just the code is a little bit different.
Interesting. No, that makes a lot of sense. And it does, I don't know, it speaks to this, like, belief or assumption that I have that, like, molecular phylogenies are probably a bit more, they're less subjective than phylogenies that are based on morphological traits from the fossil record. But it is nice to hear, I guess, we're not the only ones who have some subjectivity in that.
I think it's maybe a little less, but there's still those threshold arguments of when you call something as different, and sometimes it's statistical magic that is like Lito loves to go off on these because they're not necessarily the appropriate statistics that are being used for comparison sometimes given your data sets. But with reviewers, you're not necessarily the one who's an expert on statistics, and so are you really the one to make the commentary on it? So it's not always straightforward, but at least it's I think it definitely seems easier to me than the fossil side of things, because like you've said, like it's not even necessarily a large number of samples, and you can't necessarily go get more samples when you want to, and there's just a lot of limiting factors.
Oh boy, we're going to talk about that in our fossil paper today, Bolen. Prime example of that. Holy cow!
Yeah, it's
oh boy,
definitely one where the renaming too of like going back. It's cool, but it's like, oh man, it's like I wish we didn't have to do it. Like keep going back. We just had these to begin with and threshold things differently. But like you said, sometimes it's the competition too for naming something is almost takes over. Yeah, so the rest of the paper is really cool. They started doing these stains as well, and why this is interesting is usually a lot of these antibody stains are not cross-reactive, so they work in one particular species, and then that's kind of it. Like, it doesn't work in another species because they're different enough where it doesn't recognize the same protein, the antibody stain. And so here what you're getting is, in blue, it's making every cell that's a neuron glow, and then in red, there is this RNA for this gene that they think is producing that particular neuronal type. And so it's a way to mark your neurons of interest, like, are they present in these animals? And so what you're seeing is, in this melanogaster, they're seeing very high numbers of the red signal, and in Yakuba, they're seeing very, very low numbers of the signal. And it's cool as they did it 2 different ways. And also, Chad, the replicate number, like, I'm sure, Dan, you'd be like, man, if we had 10 dinosaurs, wouldn't that be great to make that conclusion versus, like, part of one.
Yeah.
And it's neat that they tested the other species as well, and this is why I kind of started like nerding out over the paper, because they tested in those 2 sisters, the Yakuba and Santamea, which we know don't do the particular sound that they're interested in looking in, and then they tested those flanking species. So Drosophila tesserae and erecta. Tesserae is a little bit weird, it looks like, based on the data. It's not statistically different, but the erecta is. And so back on the tree. Oh. They were showing that Tesserae and Erecta do have this sign. They're the orange wave to their song, versus it's the Santame and the Yakuba don't. But by eye, if you look at it, the Tesserae looks a little bit different. So if we zoom in on the... Oh, come on. Oh, I guess we're at max zoom. Yeah. If we zoom in, those little curves look a little bit different between the Tesserae and Erecta. Maybe that's the cause of the variance, and that's a little bit of what they suggested could be driving some of those differences. Maybe it's like... It's only different because of that wave, and there's like some almost an intermediate stage. Like maybe, you know, the Tesserae started to have a little bit of a difference form or some selective pressure, and then it really came to fruition in Yakuba and Santamea.
Interesting.
Maybe.
A little bit slightly off topic here, or different direction, but I'm wondering, how do you create those wave graphs like that, showing the frequency of the sound and amplitude and everything else? How do you like standardize that? I'm wondering, is there like a software that everyone uses that's like a discipline standard for that, or do they just provide the info and the methods, or like I don't remember this part.
So it is a standardized way of normalizing, so you have to listen to you subtract away background audio to get the amplitude, and then there is some flavor of standardization equation that you do to make it all like equivalent. In terms of the software, that is, I think, usually standardized as well. Here we go.
Using Fruity Loop Studios or something.
Or even, I don't think they're as, it's not as easy as using Audacity. Oh, that's behind a paywall now. Yikes.
Audacity is what I use, yeah.
That's what I use too for like the stream stuff. It's like, oh, that's great.
Yeah.
Let's see. Actually, hold on. I have it in the different window. Maybe there's a little something hidden away in the preprint. Or no, does the preprint not have it? Video information. Yeah, let's see. Single cell sequencing libraries. Here we go. Song Explorer is the software.
Probably pretty standard, I'd imagine.
Yeah, so it looks like they use a deep learning interface to segment the acoustic signal. So I guess they go ahead and train the software on what is the pulse crack and what is no song and what is a sine wave. And then then they feed, that's how it is. So they feed the song to the software that now can identify each of these different regions, and it's the software that does the segmentation for them. So like when you're looking at this data of what's the pulse and what's the sine, that's because you've given a machine like the guidelines of this. You define, like, I don't know, like probably an undergrad will go in and like X hundred number of songs, like this is a sine pulse, this is a crack, this is the regular pulse, and then when you feed in new songs, it can be identified by the machine.
Very cool.
And I think that's, you're taking into account, you know, amplitude, frequency, and probably absence of peaks as well, of where they're able to define that. But yeah, it's a really cool method of data collection that you're able to add in like all this extra like layer of curation that can remove the human variance from it. Especially for these kinds of things.
Be able to make that less subjective, be able to standardize it like that. Like that's interesting. I don't know anything about like audio science, so.
The only thing I've done in the past is there's this one called the Insectivox. And so that is like it was a special recording device made for grasshoppers. And we were trying to get an equivalent made for fruit flies in the engineering department at Dartmouth. And the woman that we met, who was like the head of the engineering department, she's like this big fancy lady. And we're like, this is what we want. And she's like, you want to listen to bugs? Like, yeah. And she's like, she was like, I'm going to be straight up with y'all. I don't have time to listen to bugs. She's like, I'm very busy.
Oh, no.
But that was, yeah, it's... I know in the lab that I interviewed at Princeton that looked at the sounds that these animals made, like they had pretty much this kind of setup, like in the YouTube video where there is a couple of audio devices, there's an overhead cam, and they have that software they feed it in. I can't remember if they had already in place the deep learning so that you can blind all the experiments, but I'm sure at this point they probably do, because like a lot of these we're moving to the idea that you remove anything that can identify what the experiment is.
Gotcha.
To the observer. Yeah.
Cool. Yeah.
And Harissa's wondering a neuropeptide. So it's a short sequence, usually like a short protein sequence. It's essentially the length is what defines a full protein versus a neuropeptide or any kind of peptide. A protein is made up of a peptide chain. And so something that's just really short, we just call it peptide. The neuro part of it is that it's brain specific. So imagine these really, really small molecules that when they change position can activate or deactivate certain things in one's brain. And they're fast acting too. That's a function of just their size. Let us know, Harissa, if that makes sense. Yeah, it's a cool methodology of that they do it. And then here, in this figure E, they go through like all the different subtypes of the neurons that they're kind of looking for. And so it's like what are the TN1 neurons and how they connect. And so they have like, this is in A is a TN1 and they connect to these other kinds of neurons and you can actually mark them. And so here it's like you're marking in the brain or in the ventral nerve core, which is stemming down from the brain, where these neurons are connected to and how they communicate and if they can be visualized or not. And what it's really cool is if you're looking at it, and they nicely marked it in the Drosophila melanogaster, where in yellow that's the neuron of interest that we're looking for. And so it's kind of hard to see on the anterior, but here is the neuron in melanogaster, and then it's empty or blank in Yakuba. And so they're like, maybe this creature is missing that neuron. Which is like, that's what I like about this experiment. It's like, okay, I can also see it's missing the neuron. I'm not just being told by a phylogenetic tree. It's like, yeah, this one in Santamea is missing, and then if you look in Drosophila erecta, that dark line is much more prominent. Even a little more prominent in this tesserae species as well. Like, you're seeing these dark lines reappear, and you don't have to be, I think, a neuron expert to see something like this. Like, you see a dark line, it's there, or it's absent, and that's what's telling us about what's important here.
Interesting. Cool.
And Harissa, the reason you can't test it in humans is ethics. You cannot go in and modify anything in a human brain, because then you're going to be altering, free will, behavior, all kinds of stuff like that. So that's the most you can do in humans right now is in cells. But that doesn't tell you behavior, right? So you can look at it in a mammal like a mouse and see if it alters behavior and then infer that maybe something like that would happen in humans. But you'll never be able to test something like this kind of question in an actual human. Just, again, a lot of ethical questions come into play. And human experimentation and population changes, like that's already something that's kind of a dark spot on U.S. history with looking at underrepresented folks, who are, almost tricked essentially into being experimented on. So it's something that,
experiments and stuff like that,
very much staying away from that. And even like when you have drug trials, like that's after an absurd amount of testing in other animals, and then you have different phases to ensure that it's going to be safe. And so it's not, especially if something's going to be modifying behavior, even if it's addiction behavior, you got to do a ton of testing before that. And especially because what if you mismodify? Like, if you're only trying to change that the neurons that are behaving here, but that peptide may be doing other things elsewhere, how do you get it to a particular site that you affect and not affect the other sites? Like, it gets very messy very quickly, so you have to be very cautious when doing something like this. Hopefully that makes sense, Harissa. Yeah. And Kennedy's like, I'm not supposed to be modifying human brains. That's why I need a Twitch science. Exactly, Kennedy. That's why you're here. But you can modify fruit fly brains, and this is where I'm like, actually, this the next figure. Yeah, here we go. So this is like the winning experiment here, and this is also the one that costs. Gosh, I don't want to lie to you, Danny. This is a $30,000 experiment at minimum.
Holy cow!
Yeah, so this is.
Those are some well-paid fruit flies. My goodness.
Oh yeah. So and it's because like this is where it gets like you're gonna be like, oh come on, really? It's a proprietary kit. So what you're trying to do here is sequence the single individual cells in the brain of this creature. And so what you need to do is you need to separate out the brain into individual cells and push them through a microfluidics chamber. So essentially a chamber that has tubes that get thinner and thinner and thinner to the point where you're having single cells go through one particular point of the tube. So like you go from that like it's essentially you're funneling the cells into a single cell line. And then as those cells are going, you're going to attach these particular barcodes on each cell that make it unique. So like this barcode on this cell that's coming through, it's like I branded you, you're different from the next one, and I keep doing that to every single cell. And that way, when they come back together, you can tell them all apart because they've all been barcoded.
Okay.
And so that bit of proprietary tool is very, very pricey, and it's a very novel approach. Like it's relatively new, and the Illumina sequencing company who makes sequencers developed this tool, and so they're like, if you have to buy our sequencer, which is $1 million, and then a kit to do single cell sequencing is about $10,000.
Wow.
And the kit, Danny, does not allow unlimited experiments, as you might imagine.
Rate limited, or they
make it... No, it's right. They add in they had an extra of everything except like one of the key components, and so and you can't buy them individually, each component, because there's people out there trying to
bootleg those.
I
feel like there could be.
So that's the thing. There are labs that are trying to like make their own, but there's the proprietary components. There's a couple of like stepwise things where if you try to, when you put into the sequencer, there goes a cartridge, like a printer almost. And it's one of those where if you replace the reagents in the cartridge or try to reuse a cartridge that you've replenished, the machine's like, I don't like this. This isn't working.
So it's exactly like an HP printer then.
Yep.
You put a different brand of cartridge in, I'm going to light myself on fire.
Pretty much. And to be like, it's an amazing piece of tech, but like it's kind of shady for that reason, right? And I get people got to make money, but at the same time,
pretty exploitative. I don't know if exploitative is the right word. Shady is a good way to put it. It sounds unnecessarily, rent-seeking there.
And I don't know the background of this company, of how many failed things they had, right? And like how much of a loss they incurred developing the tech, all these different things, and like what else.
Maybe there was a ton of an obscene amount of R&D that went into it. They're trying to recoup those costs or something, but I don't know. It still leaves a bad taste in my mouth.
For sure, yeah. But it's really powerful because there's no other method to get the individual cells to be sequenced. And so here, what you're looking at is Bryce, sleep well, and Dr. Hubs, thank you for the follow. What you're looking for is what they did. They made a single cell suspension of the brains of these animals, and then they're trying to see if you sequence them, how do the cells look on this, like, it's called the Tisney plot, but basically like a scatter plot. And the idea is that things that are similar will cluster together than things that are different.
Okay.
And so here, the cell types are in different colors, and what you can see is in melanogaster, there's this batch of purple cells, that are not present in Yakuba.
Uh-huh.
And they line up really
Very
nice. Like
cool.
if you were to overlay these two graphs, you could very easily
Yeah.
see like the red is still in like that 10 zone for both species,
Uh-huh.
and then that purple batch of cells is missing. And so that's how they can get to like we've seen it like it looked like the neurons were gone in the images, but here you're not looking at morphology, you're looking at the RNA profile of every single cell in the brain.
Gotcha.
And by doing that comparison, it's like, okay, well, what's different? What's the same?
Yeah.
And it looks like this batch is totally missing.
Interesting. It's sort of kind of tied all together for everybody watching. Where the rubber meets the road, what does this actually mean for fruit flies and their songs? Like, how does this apply to that?
So this, I think, further solidifies down, or at least is very close to suggesting that without these cells, you cannot do this element of the song.
Gotcha.
And so
like, that ability to sing that part of the song is tied directly to those brain cells that are coded there. Really interesting stuff.
and they do it like a nice, like a second kind of image labeling too to further highlight the blues here. And it's like, this is looking at a particular gene. So here is just cell type. Like I want to cluster everything by what's similar and what's different. What you can do is when you have that graph, you can impose an added layer of data on it. And that added layer of data is a gene of interest. And so you could, like Dan, you could be like, what's your favorite gene ever? You'd be like, I think that this gene is the thing that's responsible for this difference. And so it's going to be one of two effects, right? Either you're wrong, and there's going to be colored dots that light up everywhere, or here it looks like they're right, where this 17D3 gene is very, very highly expressed in that little cluster of cells that's in melanogaster but absent in Yakuba. And so it's not perfect, right? There's a couple of blue dots out here. There's a few blue dots, like in the Yakuba one, but it's definitely in all those cells that are in that little square that are missing in Yakuba.
Gotcha.
And so that's how they're trying to be like, well, here are the neurons that are missing. Here is the gene that's different, that's contributing to the formation of those neurons. And then this was my favorite experiment, is that they go in and they actually remove that gene from the melanogaster fruit fly. So now it's like, okay, if this is what is responsible, if we take out this cell type, by getting rid of this gene,
we would still be able to produce those songs. Yeah.
And it turns out
that's really elegant. Yeah.
Yeah, then that Drosophila melanogaster that had that cell type and had that gene, if you get rid of that gene, all of a sudden it collapses down, and so you can see it right here. So this is the TN number, and as we're looking across with this, you're having a marker, this mirror GFP, and all that does is mark the cell types, and then they put in the Yakuba copy. They couldn't put in the Yakuba copy of the gene, and it gets rid of that behavior, which again, I like. That is super freaking cool that something like that can happen.
Yeah.
And
really neat that you can demonstrate it like that. That's pretty special. Holy cow.
Yeah, it's really, really cool. And I thought it was really, really elegant too, that you can take this Yakuba copy and put it into the Drosophila melanogaster and change like the amount, this percentage here of those neurons. And then if you do the opposite too, if you put the melanogaster copy into Yakuba, they start gaining those cell types back. So
that's really neat.
They can't then do the song, which kind of makes sense, is because presumably like there's other factors at play of being able to do the song, like even like behaviorally hardwired, right? But that gene is responsible for the neurons. If you get rid of the neurons in a fly that does the singing, it gets rid of that part of the song, not the entire song, just that one part. It's already missing in Yakuba, so kind of everything together, I think, very elegantly describes how this may have evolved through the loss of the expression of this one gene that got rid of those particular neurons that got rid of that song element. And so I don't know. I thought it would be really exciting and folks would enjoy it because we talk about evolution so much between our streams that this is like a molecular pinning of how this evolution may have happened between these creatures.
Really cool, and you can see how this would cause a speciation event too, like a mutation to be able to get rid of that cell type, which gets rid of the ability to sing that kind of song. You could have sympatric speciation like that, where suddenly this population just splits in two because ones with that mutation are just not singing to the other ones, and they're not breeding anymore, and so you just get two distinct populations that eventually become different species. That's really cool.
And I do wonder, like, how something like that happened. Like, what was the driving force for that change in song, right? Like, what was the selection pressure? Or was it just random mutation, right? Like, that could happen too. Like, and why that was successful, you know, that's an interesting one too, of like why something like that would go well versus it wouldn't. Like, what in the population made it spread? And there's not really any of that in the paper of like...
I would have loved to have seen more discussion of that, because that's the sort of thing, as a paleontologist looking at things at more of a macro scale, I find that really interesting. Like, how does the speciation occur broader than just at like a genetic level?
Yeah, I mean, I think the only thing they've really pointed out with regard to that in the discussion was that, you know, blocking this or putting in this gene and blocking apoptosis in Drosophila yacuba did not fully restore the TN1 number to the level of melanogaster, suggesting other mechanisms may be at play.
Yeah.
So it was very, I think, anti... I guess kind of how we talked about it the other day, where there's certain phenomena that we think that is how it happened in terms of evolutionary logic, but there's nothing to say that it, like, we can't prove that that's how it happened.
Exactly that way, yeah. Huh.
So
neat stuff. Yeah.
But I thought it was super elegant. Like, I agree with you. I would have liked to see more, but I wonder if that was either a text limitation, because current bio, it tends to be really short, or if it could have been that the reviewers were like, yeah, this is just out of scope from what we want.
That would make sense. It might just not be that within the specialty of those authors. If they're used to dealing with stuff on more of a granular scale like this, talking about population level trends and stuff like that, just outside of, like you said, outside of the scope of a paper like this. You don't really need to put that in, even if it would have made somebody like me smile.
Yeah, but I think it's cool.
Yeah.
Like,
I think it's cool too.
It's something where it's like, again, I'm like, my first undergrad project was pinpointing the evolutionary origin of an immune behavior. And that that's I geeked out on, because like on the tree you could figure out where something evolved.
Yeah.
And so like that was I'm like, this is amazing. And so it's like, I think it wouldn't have been too far for me to accept that they could have said something like that.
Sure.
They also have these videos. I don't know if we wanted to see it, but this is this activation of the TN1 neuron triggered robust wing movement in sign song and melanogaster, but there was none in yacuba. And so they have this insane nomenclature here. Let's see if we can zoom. Here we go. This nomenclature is an insane genetic trick. Basically, this DSX-Gal4 is a product that's only turned on in one neuronal set called DSX. And then the LEX-A component only turns on in cells that have DSX. So every cell has LEX-A, but they only activate when cells with DSX. So you have this component only in that cell, and then you have this frit red. And so any cell that has the activated LEX-A, because it has the Gal4, can remove the stop from the frit and then activate the genetic mechanism that you're wanting to play with. So it's just a way to make it really honed in on what you're interested in to look at.
Cool.
And so it's just like a really powerful tool. And so here it's neat, like when you shine the red light. So these are like the experiments that are just like so cool because you're making a behavior happen independent of the stimulus. So here, when the red dot is on, the laser is on, and it's going to cause that behavior to start. So here, see, lights off, or no laser. They're just doing this like regular exploring behavior. Light goes on, look at the wings. They start flickering them out like we saw in that video. So like in, oh, there's out the wing, out the wing, out the wing. Doing the mating dance, out the wing the whole time. Making that song, like click, 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 click. And then red light off, the wings go back. And so
that is really neat.
Right, that's like getting at that necessary and sufficient comment that we've been chatting about, where you know, getting rid of the neuron gets rid of the behavior, but see, like here they're trying to activate it in Yakuba, and it's like this very different thing that you're seeing. There's no vibration that you're getting out of here, and it's both wings are just going out instead of the singular wing. And so it's just not working in the same way, and you're like, okay, there's something clearly different going on here. And there's the sound too. Now they added in the sound for us,
That is pretty neat.
And then there was one other supplementary video back at the very bottom. So this is if you activate that neuron in males that have that removed, this one triggers wing flicking, that's slower motion to wing vibration during singing. So it's again just a way to manipulate the behavior, and they made it they're decapitated, so you might be like, Blint, what the heck, why are they doing that? So this is getting at the question that, again, it's just a neat genetic trick of if it's the signals coming from the brain or from the ventral nerve cord, which is inside of the body. And so if you remove the head and you shine the light, if it's still causing the activation, that means it's in the ventral nerve cord, and it's not constant stimulus coming from the brain.
That makes sense, yeah.
And it's even wilder is like, well, that also means that these animals live without a head, right? And they, like, male fruit flies would try to court female fruit flies that are headless. And they live for like 24 hours.
Yeah.
And so, again, the red light will go on. And boot, look at that, you're triggering the behavior without a head.
Wow. Wild.
Yeah, and then light off, wings back. And so that, again, tells you sufficiency. Like, this is sufficient enough to trigger something like this versus, you know, being like you needing the brain to be able to do it. And there's another example of it. So I just, again, thought it was a lot of cool evolutionary biology, molecular mechanisms, and genetic tricks as well.
That is really neat. That's really neat. Wow. Cool. And I love that they had a video here. I wonder, did that video get put out with the press and everything too, or?
I haven't seen any press for this article. Was there a press release?
I don't remember you sending one. I didn't go out and look. But,
I didn't find this with a press release. I just came across.
Yeah. Which is, like, this is the sort of project, the sort of paper that if you had a video press release that went along with it, I feel like that would go a long way toward explaining it and getting more attention. And, like, it's a neat story, and there's a lot of cool visual aspects to it, and it's got really interesting implications.
Yeah.
I don't know, and I really wish that there were more video content with, you know, I wish that were more the norm in science for science communication. It's a shame that, I don't know, video work is almost regarded as this, I don't know, dark art or something like that, something arcane that, like, scientists cannot or should not do.
And I think it made it easier to understand, right, the tools that they were doing. Like,
for sure.
Light on, they did this behavior, light off, but I think I actually came across it on Twitter. I think one of my science friends tweeted that they had their paper out, and I think it was I can't remember if it was the first or last author actually at this point, but they were like, yeah, I can't remember who's tweeted it, which one was it, from the Fly World, but yeah, I was like, oh, that sounds really cool. I'm like, I like evolution and song. Let's check this out. I was like, oh, this is a neat paper.
That does, yeah, that is pretty cool. Wait, so to clarify, you're friends with one of the authors on the paper?
Yeah, I'm friends with the last and the first author, but I can't remember who
Neat.
tweeted it.
Uh-huh.
Yeah, they're at Penn.
Very cool.
So that's where we met, but I can't remember. Yeah, who it was that tweeted it.
Gotcha. Yeah. Yeah. Really cool stuff.
Hi Hogan, welcome in. How's it going? But yeah, so that was again cool evolution, neuroscience, some genetics. That was a fun paper.
It is a fun paper. Thanks for picking it. Good stuff.
And it also feels like it's finished. Like there's a future directions where they can develop more tools to look at things in the future and other evolutionary behaviors or what have you. But it feels like a complete package of this story. Like we saw there was a missing song. We saw there was a missing set of neurons in multiple ways. We figured out they were probably responsible. Thin.
Yeah. Cool stuff.
Yeah.
It does feel like a completed paper. Like it's good stuff. It's good stuff. Mini Pie, what is going on with you right now? Just bouncing off the walls.
You've had all the cat snuggles today, Danny.
I have, yeah, for sure. Especially for Mini Pie. We had to lock her out of the room earlier because she was just causing too much trouble.
I know she was getting into the lights too, right?
Yeah, she was like trying to knock over my lights, and I'm glad she's not trying to climb the back of the chair right now and jump on the top of the bookcase, because otherwise I'd have to kick her out again.
Yeah, Socks here has been unusually quiet. She's just sleeping on the floor. Usually she's getting hangry at this point and like harassing me for food. I'm like, oh, that's nice. All right. I think she might be our AC busted, so I think she's maybe just overheated or something. Hopefully, I'm hoping the guy's coming out tomorrow, so hopefully it's an easy fix. And,
nice. Yeah, we've had our AC on for the first time since I moved in today. It was finally warm enough that the AC is running, and maybe that's part of what's driving Mini Pie crazy here.
She's like, I was enjoying so much earlier on. What is this?
Yeah, but Anyway, should we go over the theropod paper?
Yeah. So
I should probably go in about 20 minutes, but that should be about as long as we should spend on this paper, I think.
Okay.
Should be pretty straightforward and pretty short. But it's we've talked before about how a lot of dinosaur science is based on like really limited specimens. This is one of those. I think I've also made jokes in the past about how the UK, Great Britain, there are more dinosaur paleontologists there than there are dinosaur specimens.
Oh wow!
I don't know. It's not entirely true, but like there are. England is where dinosaur science first got its start.
Mm-hmm.
Back in the 1820s through the 1850s. And since then, like the locus of dinosaur science has moved to other places around the world, but there's not a great many specimens that have come out of England. And sometimes even really fragmentary stuff gets named as a new genus or species, and that is one of those examples.
So it's just because we have so few creatures, and we started here, we're kind of trying to almost assert our dominance in a way.
I wouldn't say it's that, but it's like there's a specimen that a student was working on. It's like, well, let's just name it as a new thing. It's here in England, so let's do it. It's kind of more like that.
Okay.
Yeah. Did you see the actual like photos of the specimen? Did you see the figures?
Yeah. So I just as an aside, I thought the artist renderings were really pretty.
Yeah, they are, and some of these are historical. So like they're from the 1850s.
Oh, the figures themselves were drawn from the... Okay, I didn't realize that those
or they're
were...
reproduced from those originally. Some of these are photographs too. No, but I gotta swap out my earbud because this one's dying on me. Hang on, give me just a sec. But yeah, it's there we go. Looks like it's working. I like to gently tease UK dinosaur paleontology because, being here in the States, we're blessed with so many dinosaur fossils. It's incredible, especially out here in the West. Um, yeah, they're not so common in that part of the world. And so there have been a bunch of dinosaurs that have been named based on very, very, very fragmentary material. And this is one of those. It was originally thought to have been a Scelidosaur, which is like an armored dinosaur. And now it's been reinterpreted as the limb bone of a theropod dinosaur. But we're not really sure what group of theropods. It's yeah, it's a little frustrating like that. I wouldn't. I don't know. I like to think I wouldn't have named it under the same circumstances, but I'm not a paleontologist working in the UK. I don't know what that's like.
So you mean like not name it at all, but versus just like this is a dinosaur bone, and we're just gonna move on?
Yeah. You could publish a paper on something without giving it a genus or species name, without saying, yes, this is a new dinosaur and we name it, in this case, Dornraptor. Yeah, and this had been published on before, but nobody had named it. The thing is, the problem with naming a specimen this fragmentary is that this isn't a super diagnostic bone. If it were like one of the skull bones or something like that, maybe I could kind of see it, but being a limb bone like this, the problem is if you discover what could be the same dinosaur and it has everything except it's missing the hind legs, or even just this particular bone, you wouldn't be able to match it. You don't have overlap there. You wouldn't be able to say, oh yes, this new one is also Dornraptor. The less you have of a dinosaur skeleton, and you name it, the harder it is to match it up. So when I say that a bunch of dinosaurs that have been named are certainly just, the same as other dinosaurs that have been previously named, but you might not have overlap in the skeleton, or they might look different because they're at different growth stages, or they're slightly different times, or individual variation, or taphonomic variation, or various other things like that. It just gets complicated. I kind of wonder what the real utility is of giving this critter a name.
Mm-hmm.
I guess it makes it easier to communicate about this particular individual animal. If you could just say, yes, this is Dornraptor, and everybody knows what you're talking about because they've heard about it, then it's easier than just reciting the specimen number or something. But apart from that, I don't know. I'm curious to hear your thoughts on this coming from a very different field, because I know you've got questions.
So I guess I was stuck, and utility is a very good word for this, on the utility of doing this in general. Like, what are we gaining from this reclassification into a different thing? Because this is like the only bone, and so I wasn't convinced that it needed to be done in terms of making another species. Like, what have we gained from doing this in terms of like knowledge or like I understand like talking about it easier, but is that worth something like this? Because it's almost like that the splitter and lumper argument, right? Of like,
oh yeah,
is it worth just adding a dinosaur entire species just so that I can refer to this guy's specimen, or is it? And maybe I'm just being naive in coming up with that.
No, that's not a naive question at all. No, it's a great question. Part of this paper was, I guess, like reassessing this bone and figuring out that it's not a Thyreophorian dinosaur. It's not a little armored dinosaur. Instead, it's a two-legged meat-eating dinosaur. That's important, but that's something separate from giving it a new name. Yeah, there is an instinct that a lot of scientists have to name new things, to categorize them, to try and impose some sort of order on the natural world like that. That's a strong impulse. But how helpful is it always? I don't know.
I guess
this isn't like a huge high-profile journal. This isn't like a big splash paper or anything. But it's nice too that it's Dornraptor Normanized, so the species name is honoring David Norman, who's
Okay.
an English dinosaur paleontologist, a British dinosaur paleontologist. He's done a lot of cool work over the years, very well respected. But yeah, I do kind of wonder whether this name will actually get used in the literature, whether it's actually useful, you know?
I guess the first half of it maybe is really useful, right? Of like,
Uh,
let's re-examine something from the past that we've
yeah,
assumed falls into this particular group, knowing what we've
yep,
learned today, which is informative or not, right? Of what
yep,
you need, and then you reassessing old data. Like I think
uh-huh,
that's something that's at least in molecular biology not really normalized. Like
huh,
you couldn't really quote get away with a paper like that because it's not really adding,
sure,
right? Same
yeah,
reason like null data, like it's almost like a null data paper. Like we went back and reevaluated this data set, for example, and we actually didn't find anything.
uh-huh.
And so I don't know, maybe I get the feeling like maybe they thought it would be just that if they didn't add any extra. And so the extra is like,
It might be more difficult to publish on, I guess, if you don't give the critter a new name, and like if you need a publication, then yeah,
yeah,
yeah. I didn't think about it that way. That's interesting.
it's the cynic in me coming out of like,
That's
but
fair. Yeah.
but yeah, I mean that's a
Um.
commentary on the publish and perish mentality, and I again like I guess like how you're saying it, I just don't. I think the first part is really worthwhile, but I just
Uh-huh.
don't understand the naming of it. And yeah, it's like I guess it's nice to have some to honor a British paleontologist, but would could we have just named him a name after a different dinosaur?
Sure. Yeah. There are some other dinosaurs that I think have been named after him before. I think that guy was his. I think David Norman is this author's advisor too.
Oh, so then that's a little that's strange then. I did not realize
I don't
that
know if it's
connection there.
strange, but it's like it's a nice touch. But it's yeah. I'm not saying that's a reason the paper should exist.
Hmm.
That you get to name it after your advisor or whatever. But it like I don't know. I'm trying to pick out like positive things about this. There's this emphasis, like you said, of like reevaluating old fossil finds, historical specimens, looking through collections to find things. But my bias as a North American dinosaur paleontologist is like, go out and find some more specimens. um, that's hard to do in a place like England. Um, it's not as easy as it is here in the American West, certainly. So it's easy for me to say. Uh, yeah.
How much of this like this last figure kind of surprised me?
Oh, the life reconstruction of the animal right there.
Yeah.
I like it. It looks very like Jim Henson creature esque. Um, just the way that it's rendered is funny to me. It's charming.
Oh, it's very
But
pretty.
one of the wrists are like oriented in the wrong way, and like the arms look too human like. Yeah, I don't know.
That was what I was going to ask you about. Is I guess how accurate do you think this is, and do you think it was appropriate to have a reconstruction like this when so little is present? Like at what point would something like this be
Uh,
okay? Especially how you were saying that bone isn't necessarily a diagnostic tool.
yeah, so we don't know if the animal looked like this. It may have looked more like Dilophosaurus or Cryolophosaurus. It might have looked like Centarsus, Megapnosaurus, whatever. Um, it may or may not have had a feathery coat like that. Um, Probably didn't have wrists that oriented that way. It almost looks like it has an opposable thumb.
Yeah.
Um, yeah, it's like I guess they kind of needed a figure to go along with the paper for press releases and stuff. Um, yeah, yeah. This kind of strikes me as like a really early career kind of paper. It's like, you know, this is something that you do at the beginning, and you could say that you've described a dinosaur, but it's I don't know. Best case scenario, like someone finds another specimen of this animal, maybe excavating the basements for new council flats or something in somewhere near Dorset, and they they find a complete skeleton of this animal, and it matches perfectly with the specimen from this paper that you could say, bingo, that's Dorn Raptor right there. Like, called it, you know?
Okay.
But that doesn't happen very often, especially since, as dinosaur paleontologists, we definitely have a tendency to focus on the differences between specimens and not the similarities. So even if somebody did find a complete skeleton, even if it matches almost perfectly, it might still get named something else because it's like, well, you've got a brand new, beautiful, complete skeleton. Of course you're going to name it, you know?
And it's not in your legacy's best interest to name it after what already exists.
Or to just say, oh yeah, it's that we're going to call it Dorn Raptor because somebody published a paper with a single fragment of a bone, and you know, yeah. So it's not necessarily best practice. I picked this paper because I wanted to talk about your papers today. I figured this would be a short discussion, and this is like the only I guess this is the most easily discussable paper that's come out in the past week. It's been kind of a slow week in dinosaur paleontology, except for the Archaeopteryx thing that we'll talk about on tomorrow's stream.
Yeah, I'm excited to hear you talk about it tomorrow. Yeah, that's going to be a good stream. It's every stream is a good stream, to be clear, that you do, but like I'm excited. Archaeopteryx stuff is always
they
as a
can't all be
kid.
winners. I don't know.
That's not true. I'm pretty sure they're all 10 out of 10. But I like this paper though. Like it got me thinking of like, I just wonder. Like when I started reading, I'm like, I don't know why we're doing this. Kind of.
What it reminds me of is like some of the first papers ever published on dinosaurs, where there's very fragmentary material from England. It's really kind of, in some ways, a callback to those historical papers from way back in the day, and in some ways, like, I don't know how, dinosaur paleontology still is in the UK in some ways. Some things you can't change, like you can't make the fossils. Well, I guess you could make them more common by finding more of them, but it's easier said than done in the UK.
I'd also assume, maybe naively, is that there's even less landmass available for them now than there was when these maybe originally discovered. Just cause...
yeah, there's more things paved over. There are fewer stone quarries.
Yeah.
A bunch of the original dinosaurs to be published in England came from stone quarries, from places like Stonesfield in Kent, I think. Most of the important dinosaur finds that have been found, most of the important dinosaur finds that have been made in England have come out of quarries or stone pits or places like that. There's not a lot of exposed rock. England is not a desert country. And so there's not a lot of beautiful exposed rock out in the Badlands that you can just stroll over and prospect for fossil bone. If a dinosaur fossil is found in the UK, it's generally something that's out of the ordinary. Somebody was digging in the ground for some reason and then found this and then realized it was important. There's a lot that has to go right to find a dinosaur fossil in the UK, especially in England in particular.
Sounds a little bit like our chat about India, where there is more and more fossils being discovered, but it's not ideal because of the tropical climate and the rain, and you're not getting the exposed rock like we were saying. I guess it could be similar to that. That's interesting that it's, rock quarries presumably not looking for dinosaur fossils, and that we end up finding them. And naively, would that all presumably lead to more of these cracked specimens too? Because you're not maybe looking an eye out for it. You just come across it.
Absolutely. So like they'll be chopped in half. There'll be pieces missing. Because you may have already you might find those bone fragments in a pile where somebody's already hacked them apart because that's what you do in a stone quarry. Yeah.
Right. That's. I like the historical callback. Then I didn't realize that in reading it, but I'm like, all right, that's pretty cool. And I guess the first part of the importance of reevaluating some of these specimens, to ensure that with modern day, I mean, even like last week's paper, was it last week? No, the week before, where we had the paper on the new framework for what is a new species, right?
And like reevaluating the compsognathids, are they actually their own group, or are they just juveniles of other theropod dinosaurs? Yeah.
Yeah, and like that, I think that framework could be applied to other dinosaurs too, right? Of how to interpret it. So it would have, it could have been kind of cool if you would have incorporated that methodology onto this. I mean, you
it'd be tough.
Yeah,
I don't know how you would. Yeah.
Like you don't have the same, but maybe.
I like there. It does kind of get back into what you were talking about with lumpers and splitters. This desire to like name new species and emphasize the differences between specimens rather than the similarities. That is kind of the same mindset that might also lead you to name something based on superfragmentary material. But yeah, I don't know. It's not like this really harms anybody. Maybe it'll be a little bit annoying to have to like sort this out. If anybody finds a similar critter in the same geological formation from the same fauna, and then like, oh, is it the same thing or is it not? Like, that's a little annoying. But grand scheme of things, this doesn't do any harm.
I guess.
And it will certainly make the people of Dorset happy to have a new dinosaur that's just been named. Everybody loves dinosaurs, and like this will probably be a science PR win, I suppose. How could it not be?
I mean, yeah, that's true. Usually people get really excited about just anything dinosaur, right? And that's.
If this inspires like even one kid to go into the sciences, because like, oh wow, a dinosaur from my hometown. That, I don't know. That's neat to think about.
Yeah.
That makes me happy.
I guess I was thinking with that other paper, almost as a way of like, first part of the paper stays the same here. We show that it's a different dinosaur and it doesn't fall into the original group that was proposed. And then the second part is, here's all the data that we would need in order to really be sure of where this falls based on this like model. And because we only have this one sample of this one bone fragment, we don't have enough data to really say if it's a new specimen or not, but we could hypothesize that maybe it is. And then it's
it will be, that's the thing, is it will be a new species. So like this is a new species, they can confidently say that, because there hasn't been, to my knowledge, another individual, like another genus or species of this family of dinosaurs found in this formation.
Gotcha.
Like coeval with Scelidosaurus. So like, it is new. It's just crummy that it's based on something that's so fragmentary. Because if anybody finds even a complete skeleton that just happens to be missing the this particular bone, then like, oh shoot, you don't have overlapping material. It is new, and I'm not saying it's not. But I wish there were more of it. Holy cow.
I guess like the threshold of calling it like we know it's new, but at what point is it
Can you
new?
name it? Yeah.
Exactly, like complete enough to give it a name where, like, actually, like you said, right, if someone finds the complete specimen, I mean, is it really gonna match to this, even if it's the same species? Like, it might be different. It probably is gonna be different enough on this one bone where you're like, it looks different, or like age, right, coming into factor. So yeah, true. I mean, I hope they find a more complete one.
That would be really, really cool. And it's possible. I mean, the dinosaur from the same fauna, Scelidosaurus, that's the most complete dinosaur ever found in England. It's, like, complete. Nose to tail. Almost every bone is represented. And this critter got washed out to sea and was deposited, it was buried in ocean bottom sediments at the time. That may or may not be true of this critter as well. I kind of didn't really have time to read through the full paper. I just kind of skimmed it, so I forget the depositional setting there. But actually, it might not even be known, because this thing was dug up back in the 1850s. There might not be good information on where exactly it came from. Yeah. But yeah, that's just kind of frustrating, I suppose.
Yeah. But it's cool though to talk about it because we identify why it's frustrating, right? And like what we do differently and what information we like extra and why
Yeah.
it's not an ideal situation.
It's very far from ideal. So it's an articulated knee. The original specimen was a femur and a tibia stuck in articulation, so they were together, which implies that there was more of that animal there.
Mm-hmm.
If you have two bones in articulation, like there would have been more of this animal. But I think the tibia has since been lost, and all they have now is the femur. So like over the years since the 1850s, that tibia somehow got broken off and lost. You know, I wonder, maybe, I guess there is a chance that like more of this creature could exist in somebody's private collection somewhere, and it could be reunited. Stranger things have happened in UK dinosaur paleontology for sure. Maybe that'll happen someday, and then, you know, a more complete specimen that gets donated after somebody's death to a museum, maybe that'll bear the name Dornraptor for forever thereafter because, you know, because this paper was published. But I don't know, you know. Yeah.
Yeah, it's neat. It's still like I like the lessons learned from it and what we can garner and
Sure.
like, you know, having it be a discussion point of what amount qualifies it as a species, I think is important to have, and it's informative to have as well.
Yep. One of the quotes from the press release from the author was, "It is only by constant reappraisal of known material, often in the light of new discoveries, that we can ever truly understand the true richness of the global fossil record." And that's lofty language, but it's not untrue. Like, that's right. And I guess this contributes to that in a small way, and that's nice.
Yeah, I guess it assumes everyone's in it for the purity of the science.
Yeah, I want to believe that.
No, but like we said, you know, like there'll be like when you have the opportunity to name something, you might just want to name it because of the PR and prestige coming with it, and not necessarily because like if you were to examine this fragment and take a look and see like, well, how close is it? Maybe it's just a different age, similar enough dinosaur, and I'm not going to take the ability to name it. You know, like it still comes on that front where a little bit of that human side to it too, and that's not necessarily ruining the sanctity of science, but there's the human side of it.
Yeah.
Like you want to make your mark.
For sure. As scientists, we are all human beings. You know, we ideally are trying to work for the good of science. We're trying to put those human emotions aside to the extent that helps us with, you know, with finding out what's true about the natural world, running experiments, everything else. But yeah, at the end of the day, we are very much human, and everybody wants to name a new dinosaur. I mean, who wouldn't want to do that?
It's the same in genetics too. Everyone wants to name a new gene, even if there's a existing one in a mammal name. You want to be the one to name that insect gene, and I think that's the importance of us being here on Twitch too. Is that like we're giving that human element to the sciences? It's like it's like Danny and Belinda are just like a regular person. They're accessible. They chat to us. We have laughs. We chuckle. Do the barrel roll as earlier in the stream, right?
Yeah, that's a good point. Yeah, and hopefully we're doing a good job of that. I don't know. What do you think, Chad? Yeah, anyway, this has been a fun discussion. I'm glad we could say some nice things about this paper, and yeah, welcome to the world, Dornraptor, I guess.
I like that. Welcome to the world.
Welcome back to the world, I guess I should say.
Yeah. Correctly into the world.
Yeah. Anyway, yeah, this has been fun. I gotta eat some dinner pretty soon here. But thanks for discussing this.
Thank you, Danny, for these crossovers. And chat next week, we won't be doing this, but we're gonna try one more time before Danny leaves for the field. And if not, we'll bring it back after you get back home in, I guess, like September or October.
Mid-August. Yeah.
Because yeah, these have been definitely a huge highlight for me, and they're always a blast. And I think it kind of exemplifies what we're trying to do on the platform anyway, of bringing different voices together and chatting science. It's the best that I think it's the most unique thing that I've ever seen on the platform.
It's been a ton of fun. Thank you for for always being there to make these happen, Belinda. And I'm looking forward to the next time that we can do this. So next Wednesday you'll be traveling, right?
I'll be on a plane, down to Florida.
Nice. Well, say hello to the crocodilians down there for me if you see any alligators or crocodiles.
Maybe some fossil megalodon teeth. Maybe I'm told those are in pretty high dose down there. We're gonna try to do some fossil
very
hunting.
Cool. They've got all kinds of cool Pleistocene fossils as well in Florida, in various swamps and bayous and creeks and rivers and stuff. So
yeah,
you're gonna be collecting fossils down there?
Yep, that's part of the itinerary. There's that. We're gonna go to some animal sanctuaries. Some birding locations, the Georgia Aquarium, which should be a lot of fun to show people the animals and talk science.
I think it's the largest aquarium in the country. Yeah.
Yep.
See the whale sharks there?
Yep.
Yeah.
And there's also there's a cool research center in Florida too that studies environmental DNA of ocean creatures. So we're gonna check that out. So it'll be like 2.5 weeks of near daily IRL science programming. We're gonna try.
That is gonna be awesome. Can't wait. Shoot.
Yeah.
Also, I don't know if it still exists, but near Orlando they had this attraction called Dinosaur World with these big wooden sculptures of dinosaurs. They were kind of hokey, more than a little bit hokey, honestly. But yeah, I wonder if it's still around.
Per TripAdvisor, it's it sounds like it's there. Admission tickets, $26.
$26. Wow. Okay. Is that supposed to be Acrocanthosaurus, Triceratops? Yeah. They're not. They don't look great, but I like the idea.
Yeah. The idea is super cute. Huh.
Yeah.
I did not. I'll have to tell Lita. We'll have to consider checking that out.
Yeah. I don't know. Something to think about, I guess.
Thank you, Danny.
I would be. I don't know how much fun I would have there. I'd be one of those situations where it's like, if you don't have anything nice to say, then don't say anything enough.
No.
Stream.
That's not true. No. You make lemons out of or lemonade out of lemons all the time. You'd be like, hey guys, let's use this as a learning opportunity. And then you talk about here is this morphological characteristic that's maybe a little bit out of date. I've never heard you put a negative spin on it. No. Because you were doing IRL streams with Lordy. Remember y'all stopped and got a burger or something under this massive dinosaur statue.
Oh yeah, in Cabazon, California. Yeah, down near Palm Springs.
And you dissected what was accurate and inaccurate of that statue, and it was very very positive. And you said the meal was pretty decent for the veggie burger. So yep, there is history for you, my friend, of you being very positive about these kinds of things.
You're right. Yeah. Well, thank you for making this such a positive experience, Belinda.
Thank you, Danny.
We can pretend.
And guys, please go follow. Please follow Paleontologizing. Go follow Danny 5 days a week: Monday, Tuesday, Wednesday, Thursday, Friday, and soon to be almost every day of the week as he's doing IRL streams from the field that he'll be running.
Yeah. I'm excited.
And Danny, is there anything else that we can promo? As always, I know tomorrow's Thursday, Birds Day.
Yeah, and we'll be talking about this new Archaeopteryx specimen that's just been unveiled in Chicago. Only the 13th in the entire world, the first one in a public museum in the Western Hemisphere. So that is going to be pretty cool. We're going to be talking about how birds evolved from dinosaurs and how Archaeopteryx first sort of helped us realize that. So Archaeopteryx stream tomorrow, and then Friday we're going to be talking about Maiasaura, the good mother lizard, just prior to Mother's Day. So that'll be a lot of fun as well.
Nice. Yeah. I like the theme. I always good themes. Very nice.
Thank you. I try. Yeah.
Well, thank you very much, Danny. Please go enjoy dinner, and I'll see you tomorrow, my friend.
Appreciate it, Belinda. Thanks again.
Thank you, Danny. Bye. All right, guys, that was the lovely, the amazing, all-powerful Professor Anduza with us. Big thank you to Danny for all of these crossovers that we've been doing. It's always a lot of fun. Danny, thank you again for coming on, my friend. It's so cool to talk about these different kinds of papers and the science behind it. And I hope y'all enjoyed, y'all. Today we had dinosaur papers, not this dinosaur of this image here, although it does look interesting. But we also had the dinosaur of the new species that we chatted about while chatting about fruit fly singing and the evolution of song. So I think very fun, nice overlapping content today, especially on the evolution side of things. I hope you all enjoyed it. I know I do every single time.